Buonasera a tutti, benvenuti, grazie per essere qui in persona e ringrazio anche tutti coloro che sono connessi con la diretta streaming. Per iniziare porto i saluti del magnifico Rettore che ha avuto un impegno istituzionale improrogabile. Si rammarica per non essere qui, ma è molto interessato all'intervento, a questa organizzazione, a questo evento, perché trova in questa possibilità di avere ospiti internazionali di spicco nell'Accademia, sia nel mondo scientifico ma anche dei saperi umanistici. Ci ritroviamo qua in questa serata del Tannin Afternoon e ci ha creduto tanto che appunto ha voluto concedere il patrocinio e ci ha consentito di poter svolgere questo evento nell'aula più bella del nostro Ateneo, qua nell'aula magna. Io non vi nascondo, sono particolarmente emozionata, quindi ringrazio il Rettore, ringrazio l'Università, gli uffici che ci hanno concesso di realizzare questo evento e naturalmente gli speaker internazionali che oggi ascolteremo, e perché questo è un primo pomeriggio di altri in cui abbiamo cercato di coniugare i saperi scientifici con quelli umanistici, ampliando e quindi costruendo dei ponti di raccordo tra i vari saperi per poter far crescere anche quelle che sono le collaborazioni fra discipline e a livello internazionale. The Tannin Afternoon è appunto un evento, il primo di una serie. Qui vi riporto il programma. Abbiamo nell'ordine, abbiamo cercato di seguire un ordine che avesse un filo logico in quella che è appunto il discorso legato ai tannini, e voleva essere anche un po' un titolo accattivante, un pomeriggio di scienza e saperi, e quindi cercheremo di coniugare i due mondi, il mondo vegetale e il mondo animale, e anche non solo nello spazio ma anche nel tempo. Infatti ho qui accanto a me anche due archeologi di fama mondiale e la professoressa Abeling che invece si occupa di botanica, per cui gli interventi si susseguono con una logica: partiamo dal mondo vegetale, arriviamo al mondo animale, poi arriveremo a quello che è l'effetto dei tannini nei primati non umani e umani, e dal punto di vista delle determinazioni, sia quelli che appartengono alla storia più remota fino a quella più recente. Ma non voglio anticipare niente. Ovviamente questo è un argomento, come potete immaginare, impegnativo dal punto di vista della conoscenza, e infatti non lo faccio di certo da sola, ed ecco perché sono molto onorata di poter usufruire della disponibilità e anche dell'entusiasmo della presenza dei nostri speaker internazionali, ma non senza ringraziare anche lo sponsor che ha reso possibile questo pomeriggio, la Silva Team e il dottor Panciroli, che dopo interverrà assieme a tutti gli speaker. I tannini sono quindi dei metaboliti secondari polifenolici che appunto mediano le interazioni tra le piante e il mondo animale. Come dicevo, non è un argomento semplice da racchiudere in un pomeriggio, ed ecco perché abbiamo i nostri speakers. Quindi sono felicissima di presentarvi la professoressa Sabine Abeling, che è docente di botanica applicata presso l'Istituto di Nutrizione Animale dell'Università di Medicina Veterinaria di Hannover, il professore emerito Mark Skinner, che è professore di archeologia presso la Simon Fraser University dalla British Columbia e viene dal Canada, e il professore emerito, professor Hedges, Robert Hedges, professore di scienze archeologiche della Scuola di Archeologia dell'Università di Oxford. Come potete già immaginare, l'intervento di questi nostri speaker, i nostri special guest, è veramente un onore per me, per l'università, e celebra un po' quella che è una collaborazione pluriennale. Ve li introduco molto rapidamente. La professoressa A Abeling ha iniziato come freelancer compiendo studi sulla vegetazione e mappando quindi la vegetazione nella Bassa Sassonia, ma il suo focus, dopo aver ricevuto il PhD presso la Hebrew University, quando è ritornata in Germania il suo campo di investigazione è capire cosa gli animali e come le piante e gli animali interagiscono fra di loro. Quindi inizierà lei come speaker, come vi dicevo, e ci presenterà la sua esperienza scientifica sui tannini. Il professor Max Skinner, che appunto viene dal Canada, è un esperto di fama mondiale di antropologia forense e ha iniziato i propri studi proprio determinando quelle che erano le caratteristiche dentali, e quindi ha svolto la sua attività consulenziale e ha collegato quello che era l'effetto della dieta, quella che è la ipoplasia dentale, e domani vi do un altro appuntamento perché presenterà un libro alle ore 5 presso il Dipartimento, il suo libro alle ore 5 presso il Dipartimento di Medicina Veterinaria, e sono dei casi reali, veri, dove appunto racconta quella che è stata la sua esperienza di determinazione su frammenti ossei, per cui ha ricostruito la storia di delitti e omicidi anche nelle fosse comuni, quindi un primo caso in Canada, ma poi ha avuto la possibilità di studiare anche altri reperti a livello mondiale. Il professor Hedges si è laureato a Cambridge in scienze naturali e ha un doctoral degree all'Università di Cambridge. Si è occupato degli isotopi stabili, quindi ha perfezionato la metodica della datazione al carbonio 14 e si occupa della ricostruzione della dieta sull'analisi delle ossa dai siti archeologici, quindi ci racconta anche come alcune molecole di interesse della dieta possono essere indicative di quella che è la composizione dietetica anche di tanti anni fa. Non mi dilungo oltre, quindi diamo il benvenuto ai nostri speaker. Da questo momento terremo l'evento in inglese, quindi vi auguro un buon ascolto e una piacevole conversazione con i nostri ospiti internazionali. Welcome to Sassari. I'm really happy I briefly introduced yourselves, and now we will switch to English so that all the scientists who are listening to us, thanks to the direct streaming, can actually benefit from the presentations of our international speakers. So I would suggest to move to the first presentation, and it will be Professor Aboling Sabine, and we may sit in the first line so that we can see her presentation. Thank you, Sabine.
very happy to be here, and thank you, Maria Grazia-Cabai, to have me invited to this talk here. Yeah, PSM in the plant kingdom, plant secondary metabolites in the plant kingdom is my topic, and I would like to introduce you in the very rich and wonderful plant kingdom with particular emphasis on these plant secondary metabolites. And I would like to ask you first, what are these PSM chemically and ecologically, and where do these occur in plants? Tannins, this will be the third point. Tannins in the genus Acer as repellents, because PSM and the tannins are part of these PSM. And then at the last point, I will give you a little outlook of these topics. I hope everybody understands myself. Yeah, that's, okay, let's start. You see a plant here. It's the genus Galeopsis. It's a weed, a kind of a weed, and outwardly you don't see something special, right? But inside it's really marvelous. First, we have nutrients, we have assimilation compounds, we have primary metabolites that need the plant needs to do basic things: photosynthesis, nutrient assimilation, respiration, and growth. This is a real basic. And look what a plant has here, and inside: carbohydrates, amino acids, lipids, vitamins, and hormones. This is just the basic equipment of a plant. But there is more. When a plant gets into some problems, the stress, biotic stresses like climate or water shortage, or stress due to partners. No, there's no stress. Biotic stresses or partners could rise in a plant the production of certain so-called plant secondary metabolites. And these are particular for the plant survival or for an ecological interaction with the environment. And here I listed these plant secondary metabolites, the main groups, let's say the saponins, alkaloids, glycosides, phenols, and of course tannins. And with these compounds, the plant is, let's say, communicating with its environment, but the environment cannot understand. I mean the biotic environment. These are the herbivores or other organisms who put stress on the plant. And here I will show what I mean. For example, partners, compounds, chemical compounds for plant partners, for example, attraction of pollinators. Here I showed this flower here, and there are, for example, phenols giving this dew to the color for the flowers, which attract certain pollinators. It is a plant secondary metabolite, these plant colors. Attraction of pollinators, it's a positive let's say, a characteristic. And also defense mechanism, and it's also positive because for the plant itself, it means that, for example, there are fungi trying to colonize the plant surface, and you should invent something to get them rid of it. And for example, there are alkaloids or glycosides inside the plant. They will get a higher production in order to defend the plant against fungi, for example. And then we have also the deterrence of herbivores, and you see already here in the plant, in the leaf, that there are some holes there, and there was a herbivore. But I'm talking about the large herbivores, our husbandry animals. And before I will show this here that sometimes a plant cannot do nothing against a stressor, nothing. And evolution has overturned the plant. It was faster. For example, here these red spots are little houses of certain larvae of certain wasps. They are not, I would not say they are not parasites, but they just use the plant tissue for development into an adult wasp. But the plant, you see, cannot do anything. There are stressors, but are there really stressors? That's the question. So, and I will talk on deterrence of herbivores. And first of all, I wanted to ask, where do these plant secondary metabolites occur in plants? And here, for example, I have a poisonous plant, and you see different concentrations of these alkaloids, and you see that in the fresh plants, the concentration is almost highest. And here it is seen that the most left bars are the young rosette plant or leaves, and the middle is a shooting plant developing its flowers, and on the right it is the ripe plant. And this you can see that in the more a plant is developed, the more plant secondary metabolites it has. And that makes sense, because in the beginning the plant hasn't invested so much tissue and energy, but in the later stages it will just reproduce itself to produce seeds. And these organs are the most precious in the development of a plant, so the plant invests most of this plant secondary metabolites in the last stages. And you see as well here in silage and hay, these are conserved, how do you call products, and they were fed to animals, and the alkaloids decrease in their concentration, and this is okay for us because we would like to feed as well certain plants with plant secondary metabolites. And you see here in silage, the last stage is almost nothing, so we can relax in this kind. Okay, so that means plant secondary metabolites are concentrated in certain organs, in the most developed ones. And we have also some plant secondary metabolites that are, let's say, also part of the primary metabolites. For example, here, what do you call some amino acids. And I'm talking on amino acid that is poisonous to horses, at least, and it is occurring in the genus Acer or maple. And this is how I would like to introduce this in the world of, let's say, poisons that aren't a repellent, but the plant doesn't communicate with the horse in this case, and we will see what is coming out and how the system at least does work. Here the poisonous amino acids. They call hypoglycine A. It is written in the last line here, hypoglycine A. This is amino acid, poisonous. And here I give you a short overview on this. We have a disease in horses. It calls atypical myopathy due to poisoning with seeds of maple. And the maple contains even this hypoglycine A. And additionally, this maple contains as well phenols, and here are certain tannins. And horses do sense the phenols, and they can avoid the maple. However, only in certain stages. And this was really fascinating to research in this problem, because we had this seasonal pasture myopathy, and nobody knows why suddenly hundreds of horses died on pastures, and nobody was aware of this hypoglycine A. So there must be a reason why horses die due to a natural plant there. And there must be also a solution to avoid this poisoning. And this I will show you. Phenols and tannins are, the horse can be aware of this pre-ingestive. That means in advance of eating by smell or taste. They smell it or taste it, but this amino acid, this hypoglycine A, is post-ingestive. They eat it and then they die. And this is not the solution the evolution looks for. So there was an open problem. And I was very curious in finding out what is the background of this poisoning in horses, because normally evolution works, co-evolution, poison and deterrence works. Why is there such a poison? And here you see different species of the genus Acer, Pseudoplatanus, Negundo, Platanoides, and Campestre. And I circled the hypoglycine A occurrence in these species. And when there's some letters written, it means there is hypoglycine inside. And you see, for example, Alcer campestre has no of this hypoglycine, but Pseudoplatanus has, and also the other has. Also the Canadian maple, Alcer saccharum, has a lot of this hypoglycine A. So normally one would expect every horse to get poisoned by eating these plants. It's not that easy, but now the real story starts. Here you see a very young maple. In the very first stage, you see here the first cotyledons. Here's a seed. Here's the first cotyledons, and here you see the first cotyledons and the first adult leaves. And you see this seedling growing on a pasture, on a horse pasture. And this was exactly the situation why horses died. They ate these little seedlings and died because of this hypoglycine I, this amino acid. But why? That was a question. Here you see a horse preparing, I would say, the seedlings around the normal vegetation of grass, and you see it doesn't eat this. They are here well to be seen, and nothing is losing of fear of this seedlings. It's just concentrated on grass. So there must be a reason why this horse avoids seedlings in this stage here. Another one, the focus on the first here in the foreground. And what we saw in the field was that only these cotyledons were eaten, not the first adult leaves. Only this here. You see here a little older biting traits. And we asked, why is the adult leaf avoided? And here I give you an overview on this. We have different stages on this Acer. The young one, stage 2 and 3, and in 3 here, the first adult leaves start to appear, and here they are already developed. And you see here the related, here are the percent related to touched seedlings. That means we observed grazing horses and saw that in these stages they ate almost everything, 69%. But when it comes to this stage here, they avoided this 5.46. They ate much less of this. They spat it out. And this, I mean, to avoid over 90%, and here just almost 70%, means that there is a difference between not eaten almost all, but here, eaten almost 20%, the stages here, along with this toxic amino acid. And because the toxin concentration in this year is even higher than in that year, so the toxin itself couldn't be the repellent. And the solution was in the tannins. Here you see as well the different stages of aether, stage 1 to stage 4, and you see the total gallic acid and tannins here increasing from stage 1 to stage 5. And in the first 3 stages there's almost no tannins, and the horses weren't aware of this, so they just ate it. The seedlings in these stages are full of sugar and proteins, very tasty, but not in stage 4 and 5. And here you see the world of different compounds of tannins here. We were asking ourselves, what is the reason why horses avoid certain stages? And we saw a diversity in different tannin compounds, and here you see the different stages of this maple. Here's the seed, the young seedling, and the older stages. And you see here the single compounds, quercetin, gallol, pentosid 1, 2, and so on. Altogether we have about 300 distinct tannin compounds. And you see as well that in different stages there are also different concentrations on each of these compounds. Here's another example. Each stage has its own, let's say, composition of these compounds. It's highly differentiated between the stages, and not only the total amount of tannins, but the single compounds. And I would like to know whether the horses, for example, sense, for example, quercetin dioxide 2, or maybe this year, and I don't know. It's really amazing, this diversity of chemical compounds, only tannins here. And now an ecological, let's say, wonder appears here in this not a very attractive looking slide. You see here the single compounds of tannins, and here AP means Aether pseudoplatanus, the maple this year, and this is Aether campestre this year. And you see that they have also very different diversity or compounds of these tannin, types of tannins. And the thing is that Aether campestre has no hypoglycine, so there are no poisonings. And this question is why? Why does this Aether species does not have this toxin? And we think that instead of this toxin, they have something else to store nitrogen, because hypoglycine is a storage tool for Aether. And to see why the diversity of tannin compounds is different from that of Aether pseudoplatanus, maybe it answers why there's also different storage of nitrogen. We don't know, but Aether campestre is eaten by horses, of course, without poisoning. Yeah, and here again, the both species, because horse owners were always afraid that their horse will get poisoned also by Aether campestre, but it isn't. There's a complete different also composition of these tannins, and of course without hypoglycine. So Acer campestre, even if it's an Acer species, does not possess hypoglycine, but has a complete different pattern of tannin types here. Yeah, and this is maybe the answer why the both stages are distinct from each other. We saw also in the field that Acer campestre won't be actually eaten. When we give the horses this by hand, they accept, but in nature they don't. And here you see the answer, because Acer campestre here with the light bars has always more total phenol content than Acer pseudoplatanus in each stage. So they are really a repellent for horses and instead of Acer pseudoplatanus. And also the composition of the single compounds is different. You see here the number of compounds increasing from the younger to the older stages, and that means also something ecologically. Why is there such an increase in diversity of compounds? I don't know. So it's just interesting to follow these stages here, and it makes something with the horse. You know, it is a repellent. Stage 4 and 5 aren't eaten, so these tannins must have an ecological, let's say, message for the horse. Yeah, and there was really a solution in this problem why horses don't eat the elder stages but the younger ones, and it is a problem, of course, because the youngers aren't supplemented with tannins and both diversity and total amount. But evolution was correct. I mean, at least this Acer species gave the message to the horses, don't eat me. But the question still is, why is there a poisoning with a natural plant? Actually, one would think a horse is adapted to it. And this is, one can answer this question when you compare both Acer species, Acer campestre and pseudoplatanus. Pseudoplatanus is a forest species, and the horse inhabits open spaces, and they don't meet each other. But Acer campestre grows in the habitat of the horse, so it has a lot of more repellent. It doesn't wish to be eaten, so they have adapted to each other. And I think that planting Acer pseudoplatanus nearby horse pastures means actually that you put a horse in a habitat that has long left since evolution. Yeah, and here you see again, this is my outlook that hypoglycine as an amino acid is actually not a plant secondary metabolite, but a primary, if you would like to put it like that, because it is a nitrogen storage, but it is regarded as a plant secondary metabolite because it is a toxin. And so it causes death, and the phenols or tannins here as plant secondary metabolite, they let's say give a warning to the horse, and but this warning doesn't work in the first stages because horse and maple aren't adapted to each other. So deterrence of herbivores only works when everything is perfect, the habitat and of course the adaptation. Okay, so I think that was my last slide. So I thank you for attention. If you have questions, please. You're welcome.
Thank you, Professor Abeling, for your very nice presentation. I think we will have the time for the questions afterwards because we're going to have a round table. So I would directly go to the second presentation. That's me. So if you want, you can stay here or just sit as we were before. I don't know. If you want to stay here, it's fine for me. I like to have you close. So okay, thank you. I renew my welcome to all of you, and especially for our guests. Sorry. Who are here, and I really would like to not to do that much, but just to bridge from the perspective of a nutritionist, what the vegetal or plant kingdom speaks to animals and how animals do respond and accept this speech from the plant kingdom. So the coping to deal with some of these molecules, and we are here in the center of the Tannin Afternoon, it is something that has to be related to biodiversity. Professor Abeling was really able to say that actually we have so much biodiverse compounds, so much biodiverse stages of plants, so genetically diverse plants also in the same genus, and they also showed different profiles of plant secondary metabolites. So this is not enough because there's also a high diversity of response by the animals, and the different animals show actually different coping ability to those nutritional and anti-nutritional compounds. So my talk will deal directly to combine what plants would like to tell animals and what animals do in turn to co-evolve with plants. Why co-evolve? Because especially when we deal with primate consumers, which are herbivores, actually they have different activity, especially in the wild. So as primate consumers, they try to retrieve the benefits of nutrients that they need for energy and for nutrient requirements. That means especially the macro classes of nutrients like protein, fat, carbohydrates, and of course fiber. But along with those, a lot of other secondary metabolites are present, as Professor Abeling showed us. So, meanwhile, the plant strives and wants to live, grow, propagate, reproduce. It necessarily has to have some tools that help in doing this, all this activity, to prevent herbivory. So on the other side of the coin, animals try to get the benefits from plants. And how they do this? Well, actually, I had some questions in mind, and of course these questions are still in my head, and only some replies were given. But can animals voluntarily select tannin-rich feed? How do animals respond to tannins in the diet? And what are the consequences of tannin ingestion? So they are all levels of cognitive activity, and also on a vegetative basis, meaning hormonal, and of course a response by the animals into different mechanisms to cope with these molecules in the diet. So tannins, as said, are plant secondary metabolites and represent the fourth most abundant group of secondary compounds of the plants, following cellulose, hemicellulose, lignin, and so on. Tannins belong to a heterogeneous group of phenolic compounds, as we have seen, and the diversity belongs to the different types of plants, in the same genus to the different type of species, and in the same species to the different growing stages, and so on, to the different organs of the plant. So a huge diversity. So the animals must be very wise and clever to select adequately what they need to get energy from these plants, and of course, reducing the risk of getting poisoned or reducing the risk of harmful molecules. We can very, very generally divide tannins into three main groups. That is hydrolyzable tannins, condensed tannins and proanthocyanidins, and fluorotannins. They have different properties, just summarizing very, very generally again. And they have different also activity for the animals. Hydrolyzable tannins means that they are water soluble, they have particular astringent capability, and they have strong antioxidant effects, like for instance tannic or allergic acids. Condensed tannins are water soluble at low pH, and they are less astringent. They still conserve antioxidant activity, and we can, for instance, cite anthocyans and prophylactinidins. Fluorotannins belong to a different group of producers like algae, in particular brown algae, and they have photoprotective properties. They are antioxidant and promote wound healing. They are especially used in the cosmetics industry. All of them are so antioxidant, antibacterial, and show anti-inflammatory activity. So from a, let's say, pharmacological point of view, they're very advantageous, but this depends a lot. So, chemical properties. They're able, in fact, to bind metal, especially transition metal, like iron, or copper, or zinc, through both chelation or precipitation. And this means that from a nutrition point of view, I have to bear in mind that probably high tannin intake would reduce the iron availability, for instance, because some basic complexes are formed. They also show really active precipitating activity, meaning that they have the capability to let precipitate protein of the cell membrane, and in this they show antibacterial activity. They have some enzyme inhibition, so they are like including digestive enzymes. So according to the dose, we have a decrease in the digestibility of some nutrients. And they are complexing, they are able to complex with amino acids, may causing some deficiency. So we need to bear in mind also this. And from the feeling, from the esteroceptive activity, they produce astringency, the sensation of astringency, and they in general are bitter. So they have a particular taste in terms of bitterness. Pros. So, notably, for instance, fluorotannin compound like diacol has been shown to cross the blood barrier with potential neuroprotective activity and effects of fluorotannins, or antimicrobial activity through precipitation of membrane proteins. As said, they are also scavengers, and some are anti-proliferative in terms of have probably been associated with the proliferation, cell proliferation, and they may be useful for particular types of excess of proliferation. On the other side, contraries, they can induce potential deficiencies, especially of metals and some specific proteins and amino acids, and also they may show toxicity due to side molecules like pyrogalol, leading to severe liver or kidney failure and damage. So, tannins are diverse, they have different activity, biological properties. Animals respond differently to tannins because animals are different. So as much as tannins are different, also animal response is different. And to understand why they are so different, again, a magic word that Professor Abeling has already said is called evolution. So especially some animals can cope better with high amounts of tannins than others do, but not with all of them. So my interests were both on condensed tannins, and here's an experience with the colleagues from different university from Torino, Pisa, and which I greet because they are online. And yes, we use these low amount of prebaceous tannins, especially in slow-growing male Muscovy ducks, and we saw that at an amount of 2.5% in the diet, there was no problem in the growth of these animals. Then I was Professor Abeling, and with another colleague from the University of Perugia, we started to screen the literature about the coping ability of animals, and we saw that the different response due to the biodiversity of animals towards the tannins may be responsible to shape the landscape. Why? Especially when in nature, Professor Abeling before showed us actually the different behavior of a horse when selecting in the same field different plant species, and the clever condition that leads it to select properly, not only or avoids that plant species, but to take advantage of some parts with the minimum risk of some probable plants with high phenolic compounds. And one of the reasons why, especially, animals can cope with high tannins, especially hydrolyzable tannins, is their salivary secretion. So salivary secretion is different if we deal with herbivores and between herbivores if they are grazers or they're browsers. And there's a different salivation if we deal with omnivores, and totally different, again, salivation if we deal with carnivores. Meaning that the activity of saliva is not only to lubricate the bolus, helping the chewing and swallowing the bolus and so on, and started and digestive activity, but also to produce some protein that are able to, let's say, be used instead of other with higher nutritional potential for the animal, which are the proline-rich proteins. Proline-rich proteins are based on a particular amino acid which is considered to be non-dispensable. So it means I use a non-dispensable amino acid to produce those proteins capable to have a high affinity to bind strictly to tannins and to limit their capability to bind other proteins where other amino acids could be instead much more important because essential to the animal to produce own proteins. And actually, if we just have an overview of the feeding behavior of animals, especially of herbivores, we find on the left side the browsers and on the right side the grazers, meaning that browsers normally face higher amounts of tannins in their diet and in general of polyphenolic in their diets, simply because the selection of vegetation lead them to have a higher content of plant secondary metabolites due to the composition of the diet. So, for instance, we have leaves, twigs, buds, where the amount of tannins is higher if compared to the amount of tannins in the grass, where the grass is the basic of the diet for grazers. So this means also that as these animals co-evolved in their own habitats, and in the between you find intermediate feeders that can reasonably shift to be more browsers or more grazers according to the vegetal composition, then you realize that also the capability of coping that high amount of metabolic secondary metabolites is higher in browsers rather than in grazers. They are much more exposed. So, actually, diet selection is a crucial decision that each individual has to make every day in order to survive, especially in nature, when you have to consider how much possibly toxic or harmful compounds are in the diet beyond nutrients. So, herbivores respond to plants and vice versa. Continuum of mammalia herbivores based on feeding niche. Browsers, which consume foliage from shrubs and trees. grazers which in general consume much higher amounts of grasses. Sorry. What's in the meaning of the response between browsers and grazers? Yes, actually, browsers select energy-dense feed, show a high feed selection capability towards energy-dense feedstuffs. On the contrary, grazers possess smaller liver, larger rumen, and longer intestine. So generalistic feeders, to be against, let's say, selective feeders, have to overcome a high amount of volume but less density or less rich, tannin-rich diets. In the middle, of course, intermediate feeder. So this has a different morphological adaptation of the gastrointestinal tract, but also secretive adaptation of the gastrointestinal tract. And salivary production is one of the first line of defense against the protein precipitating activity of tannins by the use of proline-rich protein in the saliva to limit the sequestering of complexing of other important amino acids for the animals. So morphology tells the function, and anti-nutritional compounds can be successfully, for instance, detoxified in the liver and in the rumen, especially some alkaloids by ruminants. And this is one of the first experience we had while I was there in Hanover. So I take the chance to greet Professor Campus, who is following us from Hanover, and in his institute by that time, now it is ruled by Professor Christian Vischer, which I greet, we started to feed pigs with acorns. And we were astonished of what we found, because the dramatic difference between the parotid glands of the pig fed the acorn combined diets in the top of the image was very, very different from the same piglets fed for the same amounts in the diet and for the same periods if compared to those fed the conventional diet. Meaning that the adaptation to the presence of tannins in the diet from acorns actually led to a hypertrophy activity of the salivary glands, but also in the composition of saliva, where the increase of proline-rich protein in saliva also was seen as a direct response to tannin in the diet, in the acorns. So, following this first experience, increase of proline-rich protein in parotid gland, we actually focused on the bilateral parotidomegaly, which actually turned to be a hypertrophy induced by the acorn consumption in pigs, dependent on individuals' age but not on intake duration, because actually the response of the salivary production is immediate. It was enough, just 24 hours of administration, to have a response of the parotid gland. So it was immediate. So as a first line of defense, it turned to be not on intake duration, but surely on individual's age, meaning that as the animals grew, the response was even higher. And this is also reasonable, because younger animals probably have a less diverse diet than adult animals in nature. So, considering the effect of astringency due to the tannins in the diets, actually the mechanism is to be considered in terms of impulse of taste signal that goes by the 7th, the 9th, and the 10th pair of cranial nerves. And due to this, the signal reaches the cortex and makes the individual aware of the taste, of the bitter taste. And the feeling of astringency, that is a typical sensation that you have due to the protein precipitating activity of tannins. So in the mesencephalus, we have the salivatory nuclei, where the triggering from the external stimulus from the taste actually is able to activate and squeeze the smooth muscle around the adenomers of the salivary glands to push the saliva out in the ducts and then into the mouth to protect the mucosa from the effect of tannins. So to refresh the taste buds in order to make them ready again to detect other tastes or to get away unpleasant sensation like bitterness, actually the cortex sends signal to the mesencephalic nuclei. They reach the salivatory glands by visceral motor nerves so that the acina involuntary muscle contract and eject the saliva directly into the mouth. So the salivation is almost immediate. And also the production of proline protein starts to be in the 24 hours after administration. It was seen that there's a positive feedback due to the tannic acid, and it was mimicked by the use of isoproterenol, which has a positive effect on the signal transduction to the production of proline-rich protein with a very, very strong effect, as simulated by the tannic acid. So the signal reaches the cortex and make the individual a wearable. We have always considered that the complexing of tannins would have prevented them from being absorbed in the small intestine. And actually, this was, let's say, one of the main topics that led to this investigation. And we realized that especially mesenterial lymph nodes of piglets fed with tannins in the diet started to get darker. And here you can find on the left the mesenterial lymph nodes of a piglet fed conventional diets for growing piglets, and on the right you have the lymph nodes, mesenterial lymph nodes from a pig that received a diet combined with acorns. And actually, by elicitation under fluorescence of the different tissues, one could see also that some other molecules were actually drawn in the lymph nodes. We still have to do specific analysis on the composition of those molecules, but from a microscopic point of view, it was seen that some fluorescent autofluorescence could be seen after the intake of acorn tannins. So now I would stop my talk because it is time to leave the stage to the next speaker. So I would like to invite Professor Marcus Kinner, and I don't want to anticipate. So I would thank you for your attention, and again invite you here to go on with our evening. Thank you very much.
If you're interested, or if you'd like to, you're welcome to move closer. Pictures aren't that big, but perhaps you're happy where you are. Can you hear me without this? Or should I use it? Okay, well, I can take it it's on and you can hear it okay? Closer. I have a helper in the audience, so that's great. So I'm Mark Skinner. I was introduced as probably doing a lot of forensics and stuff to do with helping the police. Yes, I do that, and I'm going to give a talk about it tomorrow, but I'm far more engaged by topics like this, which I think are theoretically important, scientifically important. And I would really like to get across to anybody else who's listening to this and to you in the audience, particularly if you're, less than 50 years old or something, if you still want to be a scientist and you're curious about how science is conducted, I found it very difficult to work on this particular problem. I've been working on it for a long time, as you'll gather, I think. I'm going to talk about this phenomenon, point to it, these grooves in the teeth. Now that's a canine tooth, and it's called the canine tooth because it's about the same size as that in a dog. It's about yay big, okay? We all have canine teeth, ours are smaller. but they're a common, it's common to see among apes this phenomenon of grooves in the teeth. They're not supposed to be there. That's abnormal. It's a lesion, it's a pathology, if you want to use those terms. It's so common, it's almost normal. I'm going to just make a couple of points to try and help you understand this strange, strange condition, because its cause is not known. First of all, it happens when they're infants, while the tooth is still forming, when it's still deep in the bone. It's not after it erupts. And this is the young part of the tooth. The tooth starts near the tip and then just grows down in this direction, ultimately forming a root. Okay? So very young infant ape, and then you can see these successive grooves in the teeth. That's a pattern, I believe. Before I go on about that particular pattern, I want to show you something else that I think you'll agree is a pattern. Those are depressions. They vary in size, they vary in age, they vary in location. So we have a crater on the moon, 2 billion years ago that was created. One in Arizona, created 50,000 years ago, still an enormous thing. And then very similar looking phenomenon, which are just tiny impressions made in mud by raindrops. That's a pattern, widespread in space and time, and I think you'll agree it has a fairly ready explanation. We debate about hardness of things, the speed, velocity of meteors, whatever. But basically, there's a fundamental pattern, easily observed, and I think understandable. Now, this next pattern, you've seen the middle picture before. The picture on the left and the picture on the right, those are all canine teeth from apes. So an orangutan on the left, chimpanzee in the middle, gorilla on the right. The ones on the left, they're quite deep. I've highlighted them with pencil marks. That was just during the data collection phase. You can see it's occurring also in the incisor tooth as well, beside it. And so that's a pattern that's spread from Indonesia all the way to Central Africa and West Africa. That's the same phenomenon, isn't it? But it's spread over a really long geographic space. Is it the same cause? Perhaps. Now, these are fossil forms. In this case, I'm working with casts. They've been looked at with a scanning electron microscope. But basically, you can still see repeated grooves. Now, these particular examples on the left, one's from Spain, one's from China. The one on the right is a more recent hominin from South Africa, but they all show repeated grooves in the enamel. I observed this phenomenon a long, long time ago. And why is this worth studying? Like, who cares? I do, because I think it has the potential to tell us about the unique way in which human beings evolved. So I'm going to ask you, what's so special about being human? Well, we know that we walk on our hind legs, which is very unusual. We have freed hands to hold things. And besides bipedality and freed hands, we of course have language, almost unique. At least our form of communication is unique. Big brains, I'm sure you'd agree. And then something perhaps you don't think as much about is how slowly we grow up. I mean, you'll know how long kids take to grow up. You know, it takes forever, years and years and years of looking after them. Human beings grow very slowly. They potentially live a very, very long time. That is almost unique in the animal kingdom. Not quite. The fact that these grooves in the teeth of apes, both fossil and recent, are repeated, and they seem to be somewhat regularly repeated, to me means they're potential time markers. If we could figure out the cause and the periodicity of this stress which is being recorded in the teeth, we may be able to start looking at the fossil record and say, oh, this is when this ancestor humans or this ancestral ape began to take a long time to mature, and so on. Now, this is a fairly complicated graph. It's about the relationship of how big we are and how big our brains are to how long we live. This is a double logarithmic scale, which simply allows you to draw a straight line through a bunch of data collected from tiny animals, right, like mice, all the way through to elephants. This is the relationship between body size and how long animals live. And it's a great variety of animals, from small to big. We're off the graph, like way off the graph. Human beings live 4 times as they should for our body mass. We're not the biggest animal in the world, but by gosh, we live a long, long time, and we take a long, long time to grow up. Obviously, a key part about being human. If you look at the relationship between brain weight and how long animals live, we are actually still within the normal range of variation for a variety of mammals, and the correlation between brain weight and lifespan is very high. It's 0.89. So like 80% of the variation in longevity is dictated by brain mass. These are old data from Satcher from 1959, but they're brilliant. And it means that if we look at the fossil record and the fossil creature has a brain the size of a chimpanzee, it probably grew up at the same rate that a chimpanzee does. But can we prove it? Can we have a time marker in the tissues that we can use? I think those grooves in the teeth have that potential. So that's why I'm excited about them. So I've been trying to understand these grooves in the teeth ever since I started my PhD. It is humiliating to admit that that first pattern I showed you with the meteorite craters, I figured out in 5 seconds, right? Sort of. This pattern, I'm still trying after 40 years to understand the grooves in ape teeth. All I really know is that this phenomenon is really widespread in space and time in apes and some monkeys. On average, it occurs every 12 months or every 6 months. I stress on average, but definitely it's an annual or semi-annual stress event. Interesting. Now, I've been making I realized after many years of thought that I'm making 2 mistakes, big mistakes. One of them is assuming if you don't get enough to eat, it's going to cause grooves in your teeth. I'm going to talk about that more in a minute. But I made that assumption for decades. If you didn't get enough to eat, you get grooves in your teeth. The other big mistake I made was that because in many, many of these animals, the record of stress, and I can time it, is semi-annual, there must be a semi-annual stressor in the environment. Doesn't that make sense? It made sense to me at that time. Now, dental anthropologists, and I'm one of them, I've always assumed that too little to eat can cause enamel hypoplasia, and I could find hundreds of examples of that thought. So here's one. Evidence for seasonal starvation is apparent from the presence of recurrent rings of enamel hypoplasia on the incisors of the growing teeth in ancestral apes found in Spain and Eastern Europe. This is received wisdom. This one, more recent, enamel hypoplasia is among Japanese macaques. It's a monkey from Yakushima Island. Is due to, quote, food becoming scarce at certain times of the year, potentially leading to severe malnutrition. So that's dogma. That's what everybody has believed. I believed it. The anthropological studies supported that view. So the Chinese famine is always invoked as, gosh, look at all these kids that had enamel hypoplasia and they'd gone through the famine. That's got to be the cause, not getting enough to eat. That famine was the world's worst famine on record. But not all the kids got enamel hypoplasia. there's more like a third of them got enamel hyperplasia. So, is lack of food causing enamel hyperplasia? I don't think so. I have a wonderful colleague, Alan Goodman. He says, a study of Guatemalan children, this was done in the 1980s, they were very sick kids. They gave them extra food to eat, some of them. Those that had dietary supplementation had fewer grooves in the teeth. They had less enamel hyperplasia. So he says it's unequivocal. The risk of developing LEH was twice as great in the non-supplemented group as it was in the supplemented group. Clear relationship between dietary intake and the development of linear enamel hyperplasia. So that's, again, the same story. You don't get enough to eat, you get grooves in your teeth. But then Alan goes on to say, it's not clear that increased nutrition directly causes a decrease in enamel hyperplasia. The supplemented individuals experienced half the number of days ill with respiratory and disease illness. It's possible that enamel defects are more directly related to illness than to nutritional intake. I like that. So to the question, can undernutrition cause enamel hyperplasia? I'd say absolutely no. No, it doesn't. Despite being in the literature for 40 years, that recidivism is wrong, I believe. Don't try and publish, though. If you start saying that 40 years of wisdom is wrong, you're highly likely to have your manuscripts rejected or have them sent back for revisions again and again and again. This is not a popular message. And I'm distinguishing clearly between malnutrition and undernutrition. Not a distinction made by my colleagues very often. And nor by me, I must admit, of course. No, none of you will know Professor Charles Smith at McGill, but he is like the authority in the world on enamel. And I've met him and I know him, and I write to him and say, here's another question I don't understand. Dear Professor Smith, I sign myself off cowboy of science, because I'm not a scientist, I'm an anthropologist. And as my lawyer friend says, that and 10 cents will buy you a cup of coffee, that degree. I've turned, therefore, to someone who really does know what they're talking about, really does have knowledge and authority. An appeal to authority is not the best way to conduct science, but for me, it's about all I can do at the moment. And for an animal to develop enamel hyperplasia due to starvation, not enough calories, there'd have to be near death. Well, in that 80% of chimpanzees and gorillas and orangutans who are living show enamel hyperplasia. They were not near death when they were forming their teeth, no way. So it's highly unlikely that undernutrition can cause enamel hyperplasia. Now, there's another appeal I can make, and that was to some wonderful work done in the 1960s. Professor McCants, who was upset by the people starved during the Second World War in the concentration camps, he was very interested in how the body handles undernutrition. So he did some experiments with pigs. This is quite remarkable work, research, I think. He took piglets. How many kilograms does a baby pig weigh when it's born? Like 4 or 5? Like, they're tiny. He kept them at that size until they were absolutely mature. When they should normally weigh 200 kilograms, they still weighed 5 kilograms. Very careful control of their diets. And he kept the diet as fantastically good as he could, kept them alive as well, but they didn't grow. So so on the right is a normal pig, and it shows the relationship in the jaw of the teeth, upper and lower, second and third molar teeth in a normal pig. On the left, look at the size of the head compared to the rest of the body. It's really hard to stop the head from growing because those teeth have to grow. They have to be there. Enamel is really well protected against starvation. You've got to eat or you die. So this pig is growing its teeth as well as it possibly can, and I can tell you, and Dr. McCann's observed, they don't get enamel hyperplasia, despite being incredibly starved. They do get undergrowth of the jaws, their teeth are all impacted, and so on. But the enamel, they do not form enamel hyperplasia. How it is that I and my colleagues have missed this important study from the 60s, I don't know. We don't read the right literature. So, my other big mistake, beyond assuming that you could get enamel hyperplasia from undernutrition, my other big mistake was looking to find a stressor in the environment that was semi-annual. There are semi-annual events, especially if you're an ape living near the equator, because the Earth's going around the sun, and the equatorial equator is tilted like this, and so you get a double rainy season. So you do get semi-annual rains if you live near the tropics. You know, and you sit there cold and shivering. And I actually published an article about being cold and shivering and getting enamel hyperplasia. It was a completely wrong explanation, but at the time it was the best I could come up with. It took me a long time to think, not a semi-annual stressor, but semi-annual stress events are there. If they're independent annual stressors, of course, it's going to pick up 2 events per year. On average, they're going to be 6 months apart, which is exactly what I had been observing for decades. So now I'm putting it to you. What phenomenon in nature, unrelated to disease or undernutrition, can produce recurrent LEH once and often twice a year in apes living millions of years apart, from today right back to the Miocene, and living thousands and thousands of kilometers apart? It's the same phenomenon, repeated, in all the different time and space, right? The reason I'm eliminating disease as a potential explanation is that the grooves in the teeth last too long. They last 6 weeks on average, sometimes 2 months on average. That's a very long time for any disease. Most fevers come and go within 1 to 2 to 3 weeks. That's a long disease. Not 2 months, not 6 weeks. Disease is not a good explanation for enamel hyperplasia. Can do it, but I don't think it's a good one. So, I believe that the explanation, and that's why I'm here now, may be secondary plant compounds. As we've been learning earlier this afternoon, secondary plant compounds, they're common. That would explain why LEH is so common. Sorry, LEH, linear enamel hyperplasia, sorry. Seasonal secondary plant compounds, as was pointed out, are episodic. They're not always there at the same amount, or at the same time. And they're periodic, they're episodic and periodic, only affecting older nursing apes. If you look at a very young part of a canine tooth, there is no groove in it, because it has yet to be exposed to plants, because it's living entirely off its mother's milk. It's not eating any secondary plant compounds, not directly. Because I think that it's to do with plants and plant growth, that would explain why it lasts so long. These phases of coming and going of secondary plant compounds within a plant's life cycle, I don't think those are really rapid events. I think they take weeks and weeks and weeks to occur. Taxonomically restricted, we looked about browsers and grazers and so on. I'll have more to say about that, but again, exposure to secondary plant compounds varies amongst animals, and that would be why would be LEH, enamel hyperplasia, is taxonomically restricted. It would also explain why we can go way back in time. Plants have been with us for a really long time. In fact, they predate us, right? They provide the oxygen that we need to breathe. So plants go back a long time. I think it makes a consistent argument. I'm arguing that an ape exposed to something in a plant, whatever it is, can lead to enamel hyperplasia. A weakness of what I'm suggesting is this one. As I will show in a minute, there's not a lot of evidence that secondary plant compounds affect enamel formation. But I'm pretty excited about this, and I've tried to test the idea as best as an anthropologist can. First of all, if I'm right that eating plants can cause something bad to the enamel, bad for the enamel, cats, lions and tigers and bears should not have much enamel hyperplasia. In fact, so here's a carnivores, 2.5% of all the reports, only in 2.5% of meat-eating carnivores have enamel hyperplasia. In herbivores, it's 28%. That's like 10 times more frequent. That's a huge difference biologically. And you can see what the figure is for recent orangutans. 87% of them show grooves in their teeth. The fossil forms, which I'll show on the right there, some from near the equator, some from higher up in China, they too, and in this case because they're fossils, we're dealing with isolated teeth rather than individuals, still very high compared to most mammals. Why is that? Like, what is it about being an ape? I'll tell you right now that part of the explanation is that apes are very slow to grow. Like us, apes take a long time to grow up. They're forming their teeth for many seasons, year after year after year. They're still growing their teeth. Baby orangutans can breastfeed for 8 years, incredibly. And so it would not be surprising that they're ultimately going to get grooves on their teeth as they transition from mother's milk to plant consumption. I want to talk to you about some pussycats from Marion Island. It's off the southeast coast of Africa. Domestic cats aren't supposed to live there. It's subarctic. Cats were introduced in 1949, overran the island and began to eat all of the local seabirds, which offended the biologists. And so inadvertently, mice were also introduced onto the island. So you got cats and mice and these poor birds. Now I've just showed you that carnivores, and cats are carnivore, only about 2.5% of them show grooves in their teeth. These domestic cats from Marion Island are like herbivores. They've got 25% of these cats had grooves in their teeth. Now this is a wonderful natural experiment. They've now been exterminated from the island. They went out, killed them all, and did autopsies. Amazingly, they did autopsies on the stomachs of these cats and saved the teeth. As a dental anthropologist, that's like, I just want to weep at the wonderment of that occurring. So these Marion Island cats, in their stomachs, had a lot of plant matter. Yes, of course, my cat, other people's dogs, they'll eat plant matter sometimes. Dogs need to eat plants sometimes. They need to throw up, get rid of parasites, whatever. So it's not uncommon for a carnivore to eat some plant matter, but not as much as that. So if you took a carnivore and said, you have to eat plants, it's the only way you're going to survive. You got to eat plants or eat something, you're going to get grooves in your teeth. And they did. And this is a wonderful natural experiment, which as far as I'm concerned, is really strong support for my idea. Now, I'm not going to go into all the detail of how you determine timing of events in enamel, but if you were really sick as a baby, say a fever or an illness, something else, you'll have a groove in your teeth. It's not uncommon. It's not particularly common either. It depends on how well off you've been. But at any rate, enamel starts to form here, goes up a little bit this way, and then all the way down here, a bit like layering ice cream on an ice cream cone. And luckily for us, enamel forms with a circadian rhythm. If you get a thin section of a tooth, look at it under a high-powered microscope, you can see little tiny, tiny, tiny bands, which are 24 hours of deposition in the enamel. They're very, very difficult to see. What's far more easily seen are known as striae of Retzius, which take several days to form. Now, in a human being, I think in all the human beings ever been looked at, it ranges from 5 to 12 days to form one Retzius layer. Retzius layers are really easy to see. In fact, they're visible, if you look closely enough, visible grooves on the outside of the teeth. They're tiny, but they're there. They wear off fairly quickly after the tooth erupts. So these are formed pre-eruptively. And you can count the number of these layers where they crop out on the surface, and they're called pericymata. You just count them, multiply that count by the number of days that it normally takes to form a single layer, and that's your either duration of a stress event or the timing between two stress events. Because often we deal with fossils and we need to do with large samples, we cannot always know the Retzius periodicity, and so sometimes we go to literature and say, well, what's the normal range in this taxon, in this particular species? So here I'm reconstructing duration, how long the stress lasts, and the interval between stress events in a particular study that I did, looking at recent orangutans and fossil apes. So depending if whether the Retzius periodicity or number of days to form a layer, if it's between 8 days and 11 days, which in these orangutan-like animals is appropriate, the duration is 44 to 61 days. Again, too long to be diseased, in my opinion. Look at the interval between events, though. It's close to 6 months. It's not, on average, a year in these particular 3 taxa at which I looked. And I counted the number of pericymata between events. So interval 1, let's say there's 40. Interval 2, 35. And then I do the ratio of 35 to 40. It's close to 1.0. If it is exactly metronomic, it would be 40 and 40, it would be 1.0, would be the ratio of one interval to the next interval. So these are the ratios, all added up for a bunch of teeth. And you can see how cyclic this phenomenon is. It's just boom, boom, boom. So they're averaging around 6 months, and it's repetitive. Every 6 months or so, there's another stress event. I think the data is very strong, at least for the phenomenon, if we only understood the phenomenon. So I think that having 2 episodes a year, 2 episodes of stress is not, this is where I'm not going to turn to my colleagues for some help here, I think. I don't think it's inconsistent with simply saying most plants grow through a vegetative phase and a reproductive phase. They grow their tissues. They don't want to be eaten by a caterpillar. Then they grow their flowers and seeds and nuts, and they don't want those eaten either. And there are sort of these pulses of secondary plant compounds to protect those really important phenophases or plant growth stages. I think that the twice a year thing is possible. The other thing that apes do is, most apes are frugivores. They love fruit, and they will eat selectively and just strip a tree, and then they're going to move. When that fruit season, and in the tropics, fruit growth is somewhat aseasonal, they'll finish that tree, then they'll move to switch to another plant. So dietary switching is common in apes, and certainly that would produce more than one event per year. Now, so I've been trying to convince you, good case, right? That secondary plant compounds might be the explanation for enamel hyperplasia, but the evidence that they can affect enamel, almost non-existent. That's terrible for me. So we do know, the good news is for me is that there are some alkaloids from the periwinkle plant that produces, at least in hamsters, so don't laugh at hamsters, they're very important species. And also, it's inferred, quite possibly in human children as well. So vincristine may affect enamel, certainly does in hamsters. These cancer therapeutics, vincristine and vinblastine, also from the periwinkle plant, are known to cause enamel hyperplasia in children who have got cancer and are being treated with these secondary plant compounds. Colchicine, an alkaloid found in the meadow saffron, slows down mitosis, cellular replication, and is known to be toxic in honeybees, cause diarrhea in horses, and experimentally produces enamel hyperplasia in rats. There's a lignin found in coal tar, which has been used in Japan as a painkiller in pregnancy, and it's produced, in that one study, 38% of their children had enamel hyperplasia just from taking this particular kind of painkiller, and it's from the lignin. So, this is my last slide. I can make a case that secondary plant compounds cause enamel hyperplasia, but it's just a thought experiment. As an anthropologist, that's about all I get to do. And I can't do experiments with apes. They won't cooperate, and they're very slow to grow as well. I could fool around with their diet, wait 10 years for the tooth to erupt, then wait 20 years for it to die, and then make a copy of its tooth and go back to my old diary about what happened when that chimpanzee was a baby, and then maybe get an answer. But you can see how it's unethical and impractical to do the kind of experiment that does good science. Good science isn't available to an anthropologist. Hence copies of science, right? I've come here to talk to Maria Grazia Capet because I wrote to her and said, I can't get published. I can't do my research. I'm stuck. I need help. And so she very kindly invited me to come and talk about her research. She can study the amount of extra growth, hypertrophy, in the parotid gland of pigs by feeding them a lot of acorns. The pig responds very well. She's shown, as she just showed you then, that varying the age at which the pigs are exposed to tannins is a big factor. And it can stain the tissues. That's interesting for me, because if it can stain the tissues, maybe it can also affect the enamel. Maybe. And so, with Dr. Capet's help, I'm hoping to test two possibilities. One is that the tannins are binding to the proline, which is absolutely essential for enamel formation. 25% of enamel is made up of proline. The matrix part of it, when it's first forming, is proline. A good part of it is proline. Well, that proline would be just robbed away by tannins. So possibly sequestration of the proline by tannins in the diet is what's affecting the enamel forming cell. Or some secondary plant compound, tannins perhaps in particular, are just overwhelming the ability of the animal to cope. So even though pigs do very well, as I understand it, from eating tannins, and they can produce a lot of saliva, maybe you can overwhelm it. I don't know if ethically one can get away with that, but I would like to take some pigs and just say, okay, I'm glad you ate all those acorns, here's another lot, and then here's another lot, and here's another lot. And then sacrifice the pigs later, and knowing the timing of the extra tannin, look inside their teeth and see if there's, or look on the outside of the teeth and see if there's grooves in the enamel or something, some effect on enamel formation. So one more slide.
Dear Professor Skin, thank you very much for your marvelous presentation. Fascinating. What can I say? I cannot add anything, but I'm pleased to cooperate if I can provide some sample for clearing the hypothesis. Thank you so much. Then so we can move to the Professor Hedges that is going to close this evening. Thank you. the stage is yours.
Thank you, Maria Grazia. Do I take it this doesn't work to make it doesn't work to make a pointer?
No, pointer doesn't work.
Okay. I'll wave my hands around. Right, so I'm going to be talking about a project that's been running rather slowly for more than 10 years now at Oxford, with all that time with my colleague James McCulloch, and we've had numerous short-term students on this project. So, as I say, it's quite large in scope, but like much research, it's quite difficult to know which is the way up, and it's quite slow, but it's a lot of fun. So I think to understand really what the nature of this project is about, it's quite helpful to explain to you my own personal background, how I came to be doing this. I did my doctorate, my postdoc, in basically physics, chemistry, pretty well physics, and then had rather a change when I went to Oxford in 1971, where I worked, and I still am connected with, a laboratory for applying scientific methods to archaeology. And that took me into developing radiocarbon dating by what is called AMS, or accelerator mass spectrometry. Dating is obviously important in archaeology, and radiocarbon dating had been going for several years, but we were one of the first labs to essentially make our own accelerator for a new way of dating. That's me back in the 1980s with some of the equipment. And the point about this is that one could, with this new method, date samples of archaeology containing 1,000 times less carbon than had been possible before. So whereas you needed a whole gram of carbon, after this was developed, we only needed a milligram. And that made a big difference to what could be dated. And in particular, it enabled us to start dating bone, which of course is a very important archaeological material. And one could date the protein in bone, the collagen, which very often survives. So the point about this is that I'm moving from essentially nuclear physics to something that's much more biochemical. I got interested in the chemistry of bone collagen because you have to understand what you're dating and how it may be altered. And that led to becoming interested in other isotopes in collagen, particularly the stable isotopes for carbon and nitrogen, carbon-13, nitrogen-15. These come in with the diet, they're part of the diet, and therefore part of the bone collagen. But their ratio to carbon-12 to nitrogen-14 is very slightly dependent upon the source of the food. For example, marine food has a different carbon and nitrogen ratio to terrestrial food. So that got me interested in learning more about, or in providing information about, ancient diet, and by looking at these stable isotope ratios. And I was doing most of my work on that at the beginning of the 21st century. In 2008, I had the idea, which came from thinking about bone and thinking about dietary molecules and what lasts in bone, that there might be some dietary molecules that we ingest which would actually chemically stick to bone in a way which would make them stable and to be analyzed thousands of years later. So that was, as it were, a hope. I did have one example which, well, okay, I'll explain that in a minute then. So that was my hope for what I wanted to have as a research program. And when one thinks about what is involved in this, it does look rather complicated, and in fact I'm going to be taking you through this over the rest of my talk. So the first question is, what plant-derived molecules should we be looking for, should we be thinking about? Then we have to think, well, what ones might be changed during digestion, and we have to think about which ones of those might be absorbed into the bone. That's the main points, but of course we also need to know what is retained in the bone and stable over the years, and we have to think about how we can measure this. And of course all the difficulties here may reflect back upon, well, did we choose the right plant molecules to start with in the diet? So all this has to be, as it were, developed together, finally to produce evidence that shows that the idea actually might work. So I'm going to be talking about that. I also have three related, rather shorter aspects to mention. One is this question. I'll explain what I mean by untargeted analysis, but basically it's not focusing on any particular plant molecule, but the whole mixture of them. Secondly, we need to think much more chemically, I think, about what molecular types and species will bind best to bone. By bone, I'm also using the term HAP for hydroxyapatite, and also perhaps to collagen in bone. And there's a third fairly new experiment that's ongoing where we're growing osteoblasts, not clasts, we're forming bone in the lab and seeing what can be bound to molecules. And in the background has been two other publications. One I knew about when I started, which is the fact that if you use the antibiotic tetracycline, which used to be used quite a lot on humans, it actually does get incorporated into bones and particularly into teeth as they're growing. And these green lines are the fluorescence which enables one to detect where the tetracycline has gone. So there's no doubt that in certain molecules they do stick to bone and tooth tissue, at least in life. And people have seen in a few examples a similar fluorescence in archaeological bone, and they've attributed that to, if not tetracycline, some related antibiotic which perhaps came in through a plant. It's been a lot of waving of arms at this point, but at least something interesting is happening here. And there's a second study which is very interesting, published after we started, which is from bones from an Iron Age site in Israel. And these bones are actually colored red when they were discovered. And it is believed and somewhat proved, but it's difficult to be certain, but using infrared spectroscopy, that the molecule alizarin is responsible. Alizarin is the molecule that makes the paint pigment madder, called in English. I don't know what it is in Italian, but I'm sure you've used it in your history of painting. It comes from this plant here, and in fact, I think 300 years ago, there's a paper in Britain by someone who fed his dog madder, and the dog's bones, I don't know if he killed the dog or waited for it to die, but the dog's bones were colored red from the material matter. So those are clues that this idea is possible, but only really clues. So coming back to the pattern of research and concentrating on this bit to begin with, what kind of plant molecules can we use? And of course, this is the relevance to the Tannin Afternoon. I'm generally going to use the term polyphenols, which sort of covers tannins, and in a way, depends how you define things, quite a wide range of molecules. So what we want are molecules that are likely to absorb onto bone. And the only really good clues we have are these two, that are dietarily relevant and interesting, especially for ancient diets, that survive digestion and so on. And as I say, whatever we discover then needs to be thought into how the project works as a whole. Whoops. Yes, right. So I realize this is too small to make out actually what's here, but I thought I'd just show you the very wide range of possible polyphenols that do occur in diet. In fact, some of them have been mentioned. Grazia mentioned anthocyanidins earlier on, and so you have the molecular structures. Most of these are based on what is the flavonoid structure, essentially with these three rings, or two rings and condensed rings and a bond to a third. Over here I've listed some of the plants which are good sources of these molecules, but I realize, you can't read these: apricot, black tea, blackberry, cherry, lots of berry things and so on. They do vary somewhat with the kinds of molecules, but you see there's a lot of choice. These are just one example in each class. Here's quercetin mentioned, and this is in fact quercetin itself. And this is just the basic molecule, because as has already been pointed out by Sabine, you will get various conjugated molecules added on to this in the plant and possibly in the animal. We're coming closer to tannins here, and one that turns out to play quite a part in what I'm going to say is ellagic acid, which is a central part of this tannin, which is, pedunculagin, which you can imagine has something to do with oak trees. Resveratrol in wine is quite a well-known, I don't think you'd call it a tannin, but a polyphenol anyway. So that's, as I say, covering the range of dietary molecules that we might be thinking about. Of course, we can't try and do experiments on them all. We have to make choices. The next thing to think about is what happens during digestion. I'm afraid I don't know very much about this. I'll just explain the little bits which seem to be relevant and important to us, but there's a lot more to learn. Again, I realize this is just impossible to interpret diagram, but just very roughly, here we're starting with the tannin, elagitannin, and it's a way of making one of the compounds that will turn out to be the main compound I want to talk about, urolithin, A and B and C and D, which come about through what is happening in the small and large intestines. This is mainly with humans in mind, but it works, I think, equally well with pigs. So the tannin entering the stomach, there are questions. Okay, I'll keep away. Thank you. So we're reaching the stomach, then the intestines, material being absorbed in the lumen through the gut, circulating around the body, and if we're lucky, ending up in the bone there. That's the general scheme. And just to more or less summarize what I've said, plant polyphenols often are ingested when they're, in fact, those backbones I showed you are joined with glycosides. It could be other compounds. During digestion, that glycoside is often lost. So there's layers of complication here. Further, they can be altered by bacterial metabolism. And then what happens then? They may go into the bloodstream and ultimately end up in bone, or not. But that's obviously what we're looking for. So that's the aspect on digestion. What about what goes into the bone from the bloodstream? Well, these thoughts really I've already mentioned and we don't know very much about, but I will explain a bit more later. As I say, see later for our experiments on calcium adsorbate binding. I want to leave that more towards the end of my talk. But really we have to think about the molecular structures of known examples, and it's back to tetracycline, alizarin, and what we hope will come from the flavones, which I showed you earlier in the plants. There are theoretical ideas about what binds to calcium, and really, as I say, there's quite a bit of guesswork. And at this point, it might be useful just to give you an idea of the kind of methodology one needs to look at what does or might remain in bone. This is basically just a pretty picture. So starting with bone, we have to devise various methods which are effective but sensitive in getting out the molecules that might be bound onto the bone, obviously breaking it up, extracting with solvents, and finally making an analysis by LCMS. Although LCMS has been around, that is liquid chromatography combined with mass spectrometry. It actually, as I say, requires a huge amount of specialist development, which happens a lot nowadays because, particularly in drug research and so on, which has driven the technology, but it really needs to be at quite a high level. And I think there are two extra factors to bear in mind. One is there's so much calcium in there, it's quite difficult to actually deal with it. And of course, if calcium is binding onto these molecules anyway, that makes it more complicated. And secondly, although we're looking at modern bone sometimes, we really want to develop the methods for ancient bone. The modern bone is full of fats and so on. Ancient bone has none, and that forces different methods to happen. And also ancient bone has got lots of other stuff due to being buried in the ground. So, as I say, it's partly the explanation of why we're climbing up this wall rather slowly. And that's just to show you a more serious way of looking at it, which I just copied from one of the student reports we had, which shows you there's really a lot going on to, basically it's like one of those games. You start here, the bone sample, and that's where you want to get to. And you have to shake the dice to move along the board. So just to give you one idea, some idea of it, we start with about up to 2 bones of powder of, 2 grams of powder, or paste it often ends up with, extracting the lipids and then extracting the material with usually with ethanol and formic acid, and various types of filtering with molecular weight cutoff filters and ion exchange and so on. Okay, and now thinking about the evidence, the first main evidence has come, thanks to Maria Grazia, from Sardinian pigs. And this is rather old work now, I'm afraid. We've done some since, which I'll talk about, but mostly the work goes back to this point. We have looked also at other modern pigs. We've looked a little bit on pigs raised on olive pressings from Spain. We've had one archaeological project we've looked at from a site which I'll talk about, and then there are various other experiments at the end I'll mention. So, this is, I think, one of our pigs actually from 10 years ago. These are the results. So essentially this is what the mass spectrometer is seeing. These are some of the compounds which are identified. Actually, this is in the acorn supplement feed, not in the pig bone. And so the main, this doesn't necessarily correlate exactly with amounts, because the mass spectrometer has different sensitivities to different compounds, but it goes more or less. So ellagic acid, which is the main component in the tannins from oak, is also in the feed. Also, in fact, the urolithins, which is one of the things we're looking for, was in the feed. In gallic acid there, quercetin, just to keep mentioning it, not very abundant, but detectable. And this are the results from the pigs. We had 5 pigs. This is 1, 2, 3, 4, 5. This was a control from, I'm afraid it was a Spanish ham, which was alleged to be fed on acorns, bought commercially. And this was a bone from a pig bought in Oxford at the market, which almost certainly was not raised with acorns. So the pink colours are these compounds here were found in the bone. So gallic and ellagic acid, despite being in the feed, were not in the bone. But all these were, except for the Oxford bone, which had none of them, because it had no acorns, I guess. One quite interesting thing is one of the Sardinian pigs didn't have any urolithin A or B. In fact, urolithin C is not very abundant in the pigs anyway. It's mainly A and B that were important in the bones. And this pig didn't have any, but it also was different in having a very much lower growth. And the two may be related, and it would be lovely to follow that up, but that's all we can say at the moment. So we found something in a modern pig bone that relates to the diet. Exactly how or what isn't totally clear. I will come back to that in a minute, that question. Just a little bit more of the evidence. This is urolithin A, the blue, urolithin B. So you can see in these 4 pigs and in the control, the relative proportions of them. And from 2 grams, we were measuring about 1 to 5 or so nanograms of urolithin. So not a lot. And these are the molecular structures of urolithin. And just, yes, the point I said I would come back to, taken from a review about urolithins in humans from Espin et al., if you're interested in this, where the idea of eligotannins, in other words from ellagic acid, come in. The thought is that at least in humans, and I think presumably in pigs, the urolithins are produced by bacterial action in the gut rather than just dietarily. And so it's quite likely, I think, that the pigs are producing their own urolithin from the ellagic acid in the tannins, and that's then got into the bones. That's what we think. It seems to make sense that way. Urolithins have been looked at quite a bit from various points of view in humans, and this again gives you some context about the diet and what you might find this is really studied in the blood in humans. And so they've looked at rat, mouse, beaver of all things, from wood apparently. This is in Espin's review. 10 studies he mentions on humans. So it gives you some idea. Even sheep, kebasho on sheep, young oat leaves in cattle. So not commonly unless you feed them with something that contains a lot of tannins. Pomegranate juice seems to be very much the thing of choice, and I see rats were fed with oak-flavoured milk, which interesting thought. And of course red wine and so on with humans, which had been aged in oak barrels. So there's lots of interesting ideas of sources and the product of urolithin. So to summarize this point of the work, I mean, we didn't think to search for urolithins to start with. In fact, I don't think we knew what a urolithin was. But they turned up. So it's one of these examples in research where things happen that you weren't really thinking you were looking for. But in any case, the major ingredient of acorns, ellagic acid, is a source of urolithin. And so we've got something which is a kind of fairly direct, but not totally direct marker of diet, which does seem to be retained in the bone. So the point D in my scheme that I showed at the beginning, what is retained during life and remains unaltered during burial, now needs to be investigated. And the obvious thing to do is to look for polyphenols in ancient bone. Obviously going to be difficult, but that's what we have to do. And then the other two things we've looked at, I'll, as I say, come back to after that. So we've looked at one site, the site of Forcello, which is in the Po Valley. So we're still loyal to Italy up there. It was actually because we had an Italian researcher in Oxford who had access to bones from this site, so very lucky really. It's an Etruscan site, about 450 BC, and they certainly had quite a lot of pigs that were bred, slaughtered, and presumably consumed at the site. No human bones were found at the site, not buried there, but other animal bones, cattle, sheep, and red deer. And of course, one of the important things for us is that the site is surrounded, or was then, surrounded by oak woodland, and it's almost certain that the pigs would have been brought up on oak, with a lot of oak available. So we analysed, this has not been published, but we've, because really we think we need to do it, get a bit more confirmation, and the student who got this has disappeared a long time ago now. So what we have here is the amount of urolithin, and along here are the different bones. So S stands for sus scrofa for pig. So those are the urolithin levels for sus. This one, this one, this one, this one, this one, this one, this one. So all the big peaks come from pig. So some of the little, well, pretty well zero, this is sort of noise along here, come from pig. But these are unidentified animals, actually. I don't know why we analysed them. I suppose we thought it's interesting to see what they do. The O is sheep, ovis. The B is bos, cattle. And the C, which is interesting, is red deer. And when I first saw these results, I thought, ooh, this is really good. All the pigs do it, and nothing else does it, except for that red deer, and that's really rather spoiling the picture. And then, I mean, I'm not a biologist, as you know, but I looked up red deer in Wikipedia, or red deer diet, and of course they do eat a lot of acorns. So to me, that was sort of absolutely convincing proof this must work, because I was expecting it to go the other way. But it's only 1 point. So on the other hand, I think maybe we've been very lucky to get such positive results first time round. Obviously not everything is, not all the pigs are positive, but really none of the other animals are showing much urolithin, which is quite a relief. I mean, I don't think the sheep were eating acorns, of course. Related to that is the question, well, how do the results compare with what you expect from the preservation of bone? So this is a plot of the urolithin signal along here. So the strongest pig bones with urolithin are there, weaker one and some down here. So the other animals are along here. And vertically is the amount of collagen that remains in the bone, which is a pretty good measure of how well preserved. So the higher the collagen, the better preserved the bone are. So you could say this is good preservation, but we still get reasonable values even when the preservation seems to be bad. And these bones here are well preserved but don't give the urolithin signal. So what we conclude from this is that it doesn't seem to matter too much that the bones need to be well preserved, but it does look like the well preserved bones do give the better signals. But even when they're badly preserved, we're still getting some reasonable signal. So I would say that's a good case for a grant proposal for trying to get money. Summarizing, Forcello's it's our only example of finding a specific dietary molecule that's incorporated into the bone during the life from the diet and surviving that time of burial. So moving on to 3 other fairly quick little additional bits of the research path, you might say, using what we call untargeted analysis, that is to say, just using the mass spectrometer to generate thousands of peaks, which are used more as a fingerprint without trying to identify every single peak, which would take many years, even with all the modern ways of doing it that are available. So one of the interesting things is that, again, if you try and write a grant proposal saying, I'm going to stick this into a mass spectrometer and compare millions of peaks from one sample to another, people are going to say, no, you're just going fishing, and they don't, it's not very scientific, which in a way is true, but it also can be very productive. And we have only been able to do this on the modern pigs, but what we have plotted here is not looking at specific molecules like urolithin, but looking at the mass spectrometric fingerprint and trying to separate them out. This is a principal component analysis of, so the 2 principal components that in fact separates out these 3 groups. Those are the Sardinian bones, those are the ham bones from Spain, and these are the bones from Oxford. And you can see there's total distinction. In a sense, it'd be a bit surprising if there wasn't, given we know there was no urolithin there, but I mean, that's only 1 point amongst, as I say, hundreds of peaks. So that's looking really quite a powerful method for looking for differences in populations, even if one then has to do a lot of work to explain what the differences might be. And I've just illustrated that here. I mean, we could take humans and compare hunting populations with like Paleolithic with Neolithic, or all sorts of different things, even different types of animal husbandry would be an obvious case too. So I think there's a lot of potential there, but we only have that one graph I showed you so far. And the next little section, further thoughts about mechanisms of chemistry and, well, in a sense, biology of polyphenol uptake by bone. The main work that's been done has been some Japanese work on, this is alizarin again, and they could identify the chelation mechanism there and the sort of ionic salt mechanism of binding to calcium, using infrared and I don't know what other techniques, but certainly infrared in a rather technical way to do that. And this is simply outlining similar type of structural features in the tetracycline. So those are the two examples we really know work well, and I'm afraid that the other examples we don't know very well at all. They're quite difficult experiments to do, and we have tried to measure the binding in test tubes with hydroxyapatite powder and a whole lot of different compounds here, and this gives you, as it were, the amount of binding, alkaloids not binding at all, flavanones actually apparently not binding, which I find difficult to believe. We get on to phenol, flavanols, and so on. I'm not sure how useful that is. A perhaps more useful graph is one that we published, and this is ordering a whole lot of possibilities that we tested with, as it were, the percentage binding to hydroxyapatite, chlorogenic acid, which certainly is present, I think, in coffee very strongly, very high binding. This is tetracycline here. This is alizarin, and we're going from 100, that's ever down to 0 over there. I've put in, well, this is ellagic acid. This is quercetin, but quercetin conjugated as a glucoside. And in fact, if you take the glucoside off, the quercetin loses most of its binding power, which is a shame. But whether that happens in the body or not, it might do. I mean, it's just outlining the complexities, the extra information we need to know. I put in Oleeuropean here because that comes from olives, one of the polyphenols there, which is quite binding, and we're hoping, we had a preliminary look and didn't find any, but we're hoping to find that. Alizarin there, and urolithin B there, gallic acid there, just to give you a sense of, so there is a whole range of, and these are most of the flavonoids somewhere in there, a whole range of possibilities, which obviously are dietarily relevant, but you can't feed lots of animals one at a time with all those things to have a look. The other question, of course, is, and this relates a bit to what Mark is interested in, what about the role of protein? And we had a brief look with alizarin and with urolithin, treating them with myoglobin, which is a small protein, and with a solubilized collagen, which is a much bigger protein molecule. After 24 hours, what we did was to measure how much polyphenol was left when we removed, either did a molecular weight filtration, so we removed the protein, all the high molecular weight stuff, or we extracted the solvent with acetonitrile. And what we found, and I've got the results, but it's easier to just summarize, all the polyphenols were removed with the protein. In other words, the implication is that actually all the alizarin or urolithin bound with the protein and was removed when the protein was removed. So in a sense, we've got the question, do these stick to protein, or do they stick to hydroxyapatite, or do they stick to both? And I don't know the answer. I mean, I think almost certainly we know they stick to hydroxyapatite, and we know they stick to protein. But what happens actually in the bone that's growing is anybody's question. I think I'll leave those results. You've probably seen enough graphs that are difficult to focus on anyway. And my final bit of information for this talk is we've done some, which I think is very interesting. It's only just the beginning, but we've done some growing of osteoblasts, of bone cells in vitro. We found some people in Oxford who do that sort of thing. We haven't got them, the cells that is, to grow a mineral scaffold yet. That's a sort of phase 2, but phase 1 has been to actually grow the osteoblasts themselves in a medium in which we've added alizarin or urolithin A, or indeed both together. We've added that to the culture medium, and then we've made sure that we've then analysed the intracellular concentrations. So doesn't mean it's stuck to protein, and of course there's no bone in them to stick. But all the same, what we have found is that the cells have concentrated the alizarin and the urolithin inside. I mean, it has gone into osteoblasts and is present when osteoblasts should be making bone. And so it's a good start. I think one then has to say, well, and this is, again, analytical evidence for what we've done, but again, I think I'll pass that by and really say what would happen next with this. Well, certainly one obvious question is, how do the polyphenols, when they're conjugated, go into the cell as compared to the unconjugated? That's something that we absolutely need to know. I mean, the advantage of this experiment is we've got some control over what we're doing and some understanding of what the results might mean, whereas, as you've seen, when you feed something to a pig, you've got an input and an output, and you don't really understand what's gone on in between, although you may be very pleased at the result. So we'd also want to repeat the experiments when we're actually making hydroxyapatite matrix with the osteoblasts. And we could look even microscopically. One could imagine doing fluorescence spectroscopy with suitable molecules to see where the polyphenols are actually ending up in these cells. And I think this has got enough sort of science, clear science behind it, that it really ought to be fundable. So that's really, hopefully, the next step, or a next step. So summarizing really the whole thing, so far, urolithin A and B are the only polyphenol that we've found to be sequestrated into bone tissue. And in fact, as I said, it's probably a secondary metabolite. But the principle does seem to be firmly established now, that the basic idea is that yes, this does happen. What we don't know is whether the uptake by protein or by hydroxyapatite is the most important. Maybe both are. We don't know how to take account of the deconjugation or the reconjugation between the diet and the bone, what's going on during digestion. I think we have strong clues for which polyphenols would be the best candidates, but we don't really know which, in fact, practice are going to be. And, you know, as I've said there, actual experiments can produce unexpected answers. And I'd say, anyway, that it's quite difficult research, actually. Extracting and identifying the polyphenols in the bone is difficult. And if we could increase our yields from the bone somehow, I don't know whether we're extracting 5% of this material from the bone or whether we're extracting 95%. We can't, it's very difficult to quantify, or at least to quantify in the sense of knowing how much is there and compared to how much you get. if we could improve the yields and the analytical sensitivity, it would make things ever so much easier. And just a sort of final slide, really, just to give you, whet the appetite. Nearly 6 o'clock. One could look at, if this works, things like the consumption of olives, olive oil, grapes in wine, hops has been suggested with beer, hazelnuts is a very important dietary component, particularly for Mesolithic humans. Chestnuts in Japan, there's an interesting archaeology in changing from sweet chestnuts as a sort of wild resource to rice, and it's not really well understood how that happened, and it'd be very interesting to follow that. Acorns, the First Nations in America, I think possibly the acorns are a bit more palatable, but probably you could boil them better, but no doubt there are enough tannins left in there. So you see there's lots of scope for some very interesting projects there. And I've also mentioned the untargeted approach, which is a sort of different dimension to looking at specific diets. So we hope to continue the research. Thank you.
Thank you so much, Professor Hedges. I would really thank you all, but I would suggest for a 10-15 minutes pause before we go to the final round table and we open for questions to the audience. And yes, so I thank you all. We may meet again in 15 minutes from now. Thank you. So we're back. We're back also online. And thank you all for staying here. We just open now the round table and we're open for questions from the audience. So I would like to ask you if you have questions or comments about the presentations that were given today and you would like to yes, participate in the discussion, please. Oh yes, one question. Just wait, I will introduce you. Tapiwa Fui is a student in the master degree in wildlife management, conservation and control. So you have a question. Would you come closer, because so that also the question can be heard from the online people.
I think that, as I understand the question, it's whether the grooves, the furrows in the teeth are caused by a mechanical force. First of all, they're being created before the tooth ever erupts. You're quite right, though, that if you compress a tooth in its crypt, in its little home where it's forming, the tooth can become deformed. There's no evidence to support that particular explanation in this case, but mammoth or elephant teeth, that if the elephant isn't growing enough, the other molars that form directly underneath the earlier molars, they'll compress them and you do get deformed teeth, but they're not hypoplastic in the way that I'm describing.
Thank you. Before we proceed with questions, may I ask the Chief Executive Officer of the Silva team that was our sponsor today, if you would like to add and greet us for your, I would thank you of course for your support, and if you would like to add for us your point of view and maybe greet the audience.
First of all, thank you for inviting us here, and it was very nice listening to the presentations. Actually, it's nice because we work a lot with tannins, obviously, and we see some of our research in these presentations, like in the bones of the swines, to see the urolithins that we see in the plasma of the swines where we make trials to improve the health of animals, or replace antibiotics, so it's nice also that you see in the past or in history how was the consumption of tannins through fruit was always part of our diet since the beginning, and it's something that we are trying also to replicate in the modern era when we again feed to humans and animals some part of the diet that is missing now, because we are consuming in the animal especially just soy, corn without any polyphenols. And polyphenols are very important to regulate ourselves, and if you see that also in the past in the bones of thousands of years ago, you see there were urolithins. It means that we are on the right track somehow in our thinking. So it's and stimulating to see different kind of research, and also the research he was telling us, or the conversation we had during lunch was very, very, it's part of what we stimulate us, no? To have this conversation and find always new ideas. So thank you very much, all of you. And the students, I hope you study some tannins in your life, or in your, because it's very, very interesting, and it's a lot of application, a lot of things to do with them. So thank you again.
I thank you for taking part in this Tannin Afternoon for the first meeting, and yes, and for the support. So, please, other questions for our speakers? Yes, Professoressa Nuda from the Agriculture Department.
this on? Yeah. I understand the question. I wish I had an answer. I've been, to be honest, I have not given a thought to accumulation in something like muscle, mainly because it's not an archaeological object. But milk is an interesting possibility, which I don't know. I mean, that is certainly, I think both questions, yes, I'd like to find the answer. There may be work in other tissues than blood. I mean, there has been work, well, it's already been cited by Michele about, by Espin on circulating urolithins in humans. And maybe they have done biopsies on muscle, perhaps. So maybe it is already published, I don't know. But yes, it's something to think about. But I don't know the answer.
Yes, indeed, we have some studies where we analyzed the muscle of, this case was beef, and we found urolithins, we found a lot of polyphenols, just not urolithins, but a lot of polyphenols. And it was nice because it was a professor from University of Nebraska, Utah, something like that, and Bamblier. He makes similar process with what he does, but on muscle, and was a feedlot animal, feedlot plus tannin, and grass-fed. Grass-fed and tannins on feedlot, very similar composition in polyphenols in the meat, while the antibiotics and normal feedlot without tannins were almost zero polyphenols. So it was interesting to see how it was accumulating in the muscle.
Right.
And also it's interesting because it improves shelf life. This antioxidant activity of the polyphenols in the increased shelf life, the color, etc.
May I ask one question, or just speculation about the patterns of hyperplasia and animal hyperplasia? Is there any relationship about vitamin D?
I've certainly considered, I think this is on, yeah, I've certainly considered the possibility that vitamin D is a factor. There's a zoo literature which looks at the amount of exposure to sunlight and how much vitamin D the animals are getting. In the animals that I'm looking at, which are of course museum collected originally, but they're growing up in very sunny habitats near the tropics, and they're getting a lot of vitamin D, probably ample amount of vitamin D, so I don't think they're suffering in that way. I'd like to say something to Professor Hedges. He expressed a thought about tropical habitats, which are relatively uniform, not, of course, perfectly uniform, but much more uniform than at high latitudes. Whereas my animals from the tropics or near tropics, the enamel's great. And then suddenly, relatively suddenly, as far as we can tell, a groove starts to form. We look at duration of that stress only in the upper part of the groove, and then all the rest of it's recovery from stress, and we just ignore it. So when I talked about duration, I only meant the beginning part, and that was about, say, 6 weeks. But Professor Hedges is right to question the rapidity of onset of this event in a tropical habitat. And all I can assume is that if it is secondary plant compounds, the apes are switching to a new diet, a new fruit or whatever, or the animal reaches a threshold of response and is fine up to a certain amount and then can't cope, and the enamel forming cell reacts. Another distinction I probably should have made is that what may be bad for the enamel is good for the animal. So they may be actually doing quite well and responding quite well to having these substances in their system. It's just that that particular cell at that particular time of formation is sensitive to that. And so it may not be signaling something necessarily all bad for the animal.
Thank you. Any other question from the audience? Comments? Yes, a question from behind. Please come here. Thank you. Christopher O'Day, PhD students.
Yeah, thank you for the very interesting question. The first question, whether all plants do contain tannins, if I understood you right. Mm. 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 Yeah, 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 exactly. Tannins belong to the plant secondary metabolites, and they are strictly species specific. So that means each plant species has its own composition of these metabolites, and we have only big classes. I mean, for example, grasses, herbs, or legumes, or trees, or so on, to classify the amount, the total amount of tannins, for example. And there I can only say that, for example, the poacea, the sweet grasses, do have at least very small amounts of, very, very small. But as I said, it is species specific. For example, there are some species between the sweet grasses, for example, the genus Anthoxanthum or Trisetum. I mean, it's European, Northern European, not water plants, I'm sorry. They do have phenols, and they are even toxic to animals in a special context. For example, Anthoxanthum has coumarin, and it is clearly a phenol, and it can be toxic in specific situations. So that means I think there are very few exceptions in the plant kingdom. They do not have phenols or tannins. But don't ask me whether they have known. I would have to be forced to lie. I don't know. And the second question, whether were they water soluble? There are, of course, there were tannins. They are water soluble, but I don't know whether one has investigated this. Maybe my colleagues could help me in answering this question. I don't know. I'm just thinking on, because it can be that there is a practical background of this to diminish these tannins in, for example, feed for animals, to water it, for example. I know only that proteins and carbohydrates were going away when you water it. But tannins, I don't know, not sure. What about my other experts here? Did you see it's a difficult question? Thank you.
Excuse me, did you say you're studying urchins? Huh. Have you seen those sea otters that are eating urchins and it stains the whole skull purple? Well, I think certainly the whatever's in kelp that sea urchins eat. I think, what is it? Sea urchins eat the kelp and the otters eat the sea urchins, but it's staining their bones. incredibly. So, sorry, it probably doesn't help.
But in any case, your question may raise a new scientific, yes, exploration to assess the exact amount of tannins in the Posidonia oceanica. So, good, good hint. Thank you, thank you. So, I think we can stop here our roundtable. I would like to thank the speakers again, our support Silva team, and the University of Sassari for giving us this opportunity here. I would go on with closing remarks and some present for our speakers, and soon after I would like to thank the choir of the University of Sassari for being here and greeting us for this marvelous Tannin Afternoon that we have had, sharing science and now enjoying and cheering up. So I thank the University Choir, and soon after our closing remarks, we will listen to their exhibition, and today they will be led by Maestro Salvatore Spanedda. Thanks a lot. Thank you all. Thank you for coming, and thank you from remote, from online. Thank you all for joining us today in this afternoon. Bye, and to the next appointment with science. Bye-bye.
Thank you very grazie.
So before we go, I have some presents for our speakers from the University of Sassari. And yes, they are here. And this is a small present. Oh, look, right, for Professor Aboling, our t-shirts. This is for you.
Okay,
I
brilliant.
hope that the size is okay. Thank you all. So I would invite the choir to greet us, and thanks a lot. So I declare officially the Tannin